Welcome to part 3 of the notes for the Biotechnology unit, where we're going to focus a lot of attention on gel electrophoresis and DNA fingerprinting. First, let's get started with restriction enzymes, which once again are cutting up DNA molecules in cross-section. So here's a piece of DNA. Here's another piece of DNA that we're going to use because we're going to be discussing how we use gel electrophoresis to separate DNA fragments involved in like crime scene and suspect DNA analysis. And again, keep in mind that typically you see restriction enzymes are going to cut in this zigzag pattern. And between two different people's DNA, those restriction enzymes are going to cut in different locations because not everyone's DNA is exactly identical. So just to give you an idea of what that cut would kind of look like, again, a zigzag cut, cutting between phosphate and sugar along the backbone. And so when we talk about restriction enzymes cutting up DNA samples, this will be important to see when you see in just a bit when we talk about gel electrophoresis and DNA fingerprinting. A way to think about these restriction enzymes are like little Pac-Men that are going around chewing up DNA. And again, chewing up DNA in pieces to create fragments. We're not talking about cutting DNA lengthwise. You've already seen that in DNA replication. All right, so moving on to objective 11, summarizing the steps in gel electrophoresis, and explain how and why the DNA fragments move within the gel. So let's get started. I'm going to pull them up, kind of magic of technology method. Oops. Number one, we're going to say, I'm going to summarize quite a bit here. We're going to obtain DNA samples and cut all of those DNA samples with the same restriction enzyme. And this is important because if we cut with different restriction enzyme, then our results are not going to give us an accurate representation. All right, so obtain DNA samples and cut all those DNA samples with the same restriction enzyme. Step 2, we're going to load the samples into wells of agarose gel. And what that's referring to are, again, these locations here that are simply cavities in this science jello, where scientists can insert, typically using something like a micropipetter, insert some DNA. Again, it's all been cut up into fragments. So now we have small pieces of DNA that have been loaded up in here, cut with the restriction enzyme, all the same restriction enzyme. Step 3, we're going to apply an electric charge. The reason that we use electricity is because DNA itself is a charged molecule. It's negatively charged. So when we apply an electric current, it's going to move the DNA because DNA has a negative charge itself. So important to remember that DNA is negatively charged. Which means on our gel, we want to make sure that we label it correctly. And that is that the DNA is going to move towards the positive end. Just think of it like poles of a magnet. DNA is negatively charged, so it's going to move from the negative end towards the positive end. And then step 4, again summarizing quite a bit. But there you go, step 4. The fragments are going to separate by size. The smallest ones are going to travel the farthest. Let me give you an idea again just what that looks like as far as DNA bands that are moving. Fragments separate by size, smallest ones traveling the farthest. It's important to remember this too, because this is explaining the method of the creation of a DNA fingerprint. So the DNA moves because of two reasons. One, there's an electric charge. It's negatively charged, so it's going to move down towards the positive end, down in our picture. And the DNA size is important because the smallest ones will travel farthest, meaning we're going to get a separation in our gel, a separation of DNA fragments. So in other words, it'll look something like this. Let's say this is the crime scene DNA. Let's make suspect number 1 the guilty suspect. So we're going to match his DNA up exactly, or as close as possible that I can draw. We're going to match suspect 1 with the crime scene DNA, meaning it was his DNA found at the crime scene, leaving pretty much no doubt that he's the guilty suspect. Here's suspect 2, suspect 3. We're just kind of arrange these randomly. And then suspect 3, same way. Just kind of make up some lines there. And what you see here, again, go back to this idea of size, the smallest ones are traveling the farthest. The largest ones simply do not fit through the gel as easily. It's kind of like Jell-O, or maybe more like a sponge that would have several little teeny tiny pores in it. The DNA is making its way through really carefully and pretty slowly, except for these small guys here are going to travel pretty quickly. Right? So the larger fragments will be up top, smaller fragments will be towards the bottom, or in general, larger fragments will be closest to the wells where they began, because they simply don't fit through that gel as easy as these smaller ones down here. Alright, moving on to objective 12, explain how DNA fingerprints are created and used. So DNA fingerprints are created by the process you just saw called gel electrophoresis. And before we continue any farther from that, I want to show you just a couple things you could do on your own as a reference to get a better understanding of this process. This animation is the one labeled RFLP at our website, right here. And it just kind of walks you through how the DNA is cut into fragments. There's two different individuals. DNA is cut into fragments, and those fragments are going to separate by size. The smallest, or in this case they say the shortest, are going to travel the farthest, and the longer ones stay towards where they all began. In this case is on the left side. The other one I want to refer you to is called gel electrophoresis, again just found on our website. When you click on it, it looks like this. It's actually a virtual lab that's going to go through and walk you through the process, a little more complicated steps than I gave you, the process of creating a DNA fingerprint. So again, here's our process, gel electrophoresis. And how is a DNA fingerprint used? Well, let's give a few examples here. Paternity cases that you sometimes hear about on the news or crappy TV shows, daytime TV. Some other things you see on TV, crime scene investigation. In other words, determining suspect from crime scene DNA. And this can be used for a variety of reasons, but just one more thing that we'll not really talk about too much, but identifying genetic disorder. In other words, certain genetic disorders can be seen by running a gel electrophoresis, separating the DNA fragments out correctly. And I'll give you one example of that in just a couple minutes. Alright, so here we have an example of a DNA fingerprint already done for us. And this example, we're going to say, is crime scene DNA versus suspect 1, 2, and 3. And again, we want to make sure that we talk about how that gel has a charge that's been applied to it to separate these fragments. The smallest ones are furthest from the starting point, the wells. The largest ones are up here at the top in our example. So to analyze this DNA fingerprint, we would have these bands already separated out. And something to keep in mind here, these bands don't represent one segment of DNA. These bands are thousands, probably tens or hundreds of thousands of segments of DNA that are all collected at this location, distance from each well. So we have our crime scene DNA. Again, at the moment, we are assuming that we don't know who the guilty suspect is. And we've taken DNA samples from all of our suspects, 1, 2, and 3. And now we just need to figure out who is the guilty suspect. And I've given you some examples that may be a little bit extreme, but just to make some points here. You want to compare your crime scene DNA to each one of the suspects, and they have to match up perfectly, as you saw in the previous example. Every single one of these bands here in the crime scene you want to match up to one, hopefully, guilty party. So when we go through this thing and analyze it correctly, we notice that this band doesn't match up, this band doesn't match up correctly. There's missing bands here and too many bands in other cases. So right away we know this suspect is not guilty. Then we go through and analyze suspect number 2. We have extra DNA here, extra DNA here, showing that our suspect is also not guilty. And suspect number 3 matches up perfectly here at all those points, except for right here. Alright? So in this example, all of our suspects are not guilty. Police have to go find some more people to try to accuse for this, but what I'm going to do is just kind of fill in the band that would show how this person, if they were the guilty party, would look. So the purpose of suspect identification and paternity testing, we examine the crime scene DNA and compare it to all of our suspects. Suspect has to be a perfect match. It's the only way this is going to uphold in a court of law. And the chances of DNA fingerprint being the same for 2 people is 1 in like 7 trillion, which there aren't even 7 trillion people on the planet, so it's pretty reliable. The only example that would be exclusive of that would be identical twins. They're going to have, you're not going to be able to tell the difference in their DNA fingerprint. Alright, so that's how to identify a suspect with using crime scene DNA and suspect DNA. Last objective is talking about gene therapy and how they could be used to treat genetic disorders. First thing I'm going to do is give you a definition of gene transfer, and that is inserting a healthy gene into a vector, which typically vectors in these examples are viruses. So we think of viruses as typically being bad, but in these cases they're not. So inserting a healthy gene into a vector, typically a virus, to be transported to a cell with a dysfunctional gene. To be transported to a cell with the dysfunctional gene. Let me give you a picture that I stole from Wikipedia that kind of gives you a really good idea of how this works. Here you can see this vector, this kind of like strange alien-shaped thing, has got some DNA in it that's been modified. That virus is going to insert itself into a cell, release its DNA, so that the DNA is now mixing with DNA that's in the nucleus, naturally found there, and now a new gene is going to be transcribed and translated.